Well, welcome, Dr. Dasgupta. I'm Keith Deserick. I'm with the DIPG Collaborative and also Cure Starts Now here local. We're joined by Lisa Hill from Lauren's Fight for Cure. And, she also represents the collaborative, but we wanted to get with you today to talk a little bit about your grant for purine synthesis and 1-carbon metabolism.
Okay, so our lab is primarily interested in understanding the metabolism of tumor cells. And we use brain tumors, both adults and pediatric, as our model. We have a very strong DIPG project for the last 10 years or so. When we looked at the data, the omics data that's present about DIPG, we really strongly felt that there's a key missing part in it, which is the DIPG metabolome. The metabolome is now present and published for all types of cancer: pancreatic, lung, adult GBM, also breast cancer. It's not there for DIPG. And we thought that's really a missing piece. And from that, we could learn if there are novel targets in DIPG. What I understood is there's no really therapy for DIPG. And so the only thing that they have for patients is radiation, proton or photon, right?
Yeah.
So our goal ultimately is, at least in the preclinical model, to test if this drug and radiation synergizes or not, gives a synergy and works better or not. So we are going the route. But before that, we actually did this experiment in vitro. We took the DIPG cells and we exposed them to radiation, low to high dose radiation. We took all 6 cell lines and exposed them to low to different dose of radiation, and waited for. So what happens is that when you radiate cells, there's the DNA breaks, right? And the cells try to correct those breaks, right? And when they try to correct those breaks, they need these purines. These purines come from this de novo purine pattern, right? So when they're looking for purines, that's the time we add this purine inhibitor.
Okay.
And then they can get all those purines that they need. And what we found is that over a course of 7 days, we killed about 95% of the cells. In 5 out of the 6 lines. In 1 line, it was about 30–40%. But in 5 out of the 6 lines, combining this inhibitor, ATIase inhibitor, and radiation, it really killed 95% of the cells.
Hi, Dr. Gupta. It sounds like a very exciting pathway of research that might springboard us in a different direction and help find some longevity for these children. So I would like to know, on behalf of the DIPG Collaborative and Lauren's Fight for Cure, if you would accept $200,000 for your grant on purine synthesis and 1-carbon metabolism in DIPG to help benefit our kids for the future.
It will be, how can I express myself? It will be very, very helpful, very helpful, because I'm really optimistic about our project. I will be very, very glad to accept the award, and I'll be very grateful for this. And to both of you, I would like to say that this project, of all the projects I have, is really close to my heart, because anytime I see this tissue on the petri dish, it's very hard. It's very, very hard. And sorry, I don't know what to say, but
well, no, it's coming from 2 parents that have been down this path, yeah, and are advocates for the DIPG International Registry, and also some of that those donations and or our children making that sacrifice for it. Yeah, that's what we want. And what you're doing here is exactly what this was designed for, and we're tremendously appreciative of your work. And hopefully we can offer the ability to be able to let you accomplish it faster, let you accomplish it and really help to cure DIPG. So we're thankful to you. And this is part of that grant, and we wanted to award it to you.
Thank you.
Yes.
Thank you. It's devastating that we don't have an answer yet. So I will do all I can, and I'm absolutely serious about it. There are other targets that we'll also try, and we'll go all out about it. No question about it. And I'm really grateful that you found our science to be exciting and you think that it's going to be a path forward.